Hello and welcome. This webcast is part of our series on antimicrobial peptides. I'm delighted to be joined by Dr. Julio Camarero from the University of Southern California. Julio, thank you so much for being here. It's great to have you on.
Mhm.
Julio, antibiotic resistance is a growing concern in medicine. Can you share with us how your research aims to contribute towards solving this challenge?
Yeah, so we are working with a new type of antibiotics, basically peptide-based. It's a challenge because peptides, as you know, they have been struggling for the development of drugs. I mean, one of the main problems is that they cannot be, most of the times. I mean, there are exceptions and things are changing, and we are helping to do that. So they are not orally bioavailable because they can be digested by proteases. So that limits the application. So we are trying to address that. So we are using a natural product that is basically a polypeptide that actually is very interesting because it was discovered as a orally bioavailable. So it is a highly constrained peptide, so that helps the peptide to survive. And this is found in nature and actually has been used by even the way it was discovered. It was discovered as a natural extract used by the local people in Central Africa to induce labor. So if you allow me, I can introduce you, if I can share my screen with you. This is the kind of polypeptides that we're working. So this is a scaffold, okay? So it's interesting because this scaffold has loops here. I mean, this is a very interesting motif that we have in nature. They're called cyclopides, and they are usually very small, I mean, for a protein size. I mean, they're around 30 amino acids, and they are very well folded. And the thing is that they have disulfides from an assisting knot, and they are backbone cyclides. So we're using the scaffold because it has many unique properties. It has shown to be orally bioavailable. And also it has, very important, it has loops that we can use for molecular evolution. And I think, the appearance of these MDRs, right, multi-drug resistance, which is amazing. It's something that I was exposed here at School of Pharmacy, and it's very scary. So we have these strains that are actually resistant to 3 different antibiotics. And when I say 3 different antibiotics, it means 3 antibiotics with 3 different types of mechanism of action. So these are actually very, very hard to treat, right? So we have, and given to that, there is also an issue that because antibiotics are not, how to say, long-term drugs, right? So many companies actually are not paying too much attention to them, right? So because you use them for a few days, a week, maybe 2, and that's it. You just fight. So they companies are more interested. They produce more money, basically like chronic disease. So there has been a problem, I mean, developing antibiotics. And I think we made a contribution by using this, which, I mean, in this case, what we did is use one of the loops to graph a known peptide that is to be a natural antibiotic. It's a PG1 or porcine protein. And so what we're doing right now is actually, we grafted the sequence in these loops. So the cyclopeptide actually allows you to evolve the neighborhood loops to actually tailor your activity against a particular strain. And for example, when we did this, we found that by doing this PG1 grafted into loop 6 in this cyclopeptide. So we got activity against gram-negative, very good, but gram-positive was not that good, right? And as you know, gram-positives are, one of the main concerns. The Staphylococcus aureus is one of them. So what we're doing now, and I cannot talk too much because this is ongoing work. So we are actually evolving this loop, right? So to actually tailor the activity against a particular strain. So I don't know if that answers your question,
Great.
but
no, great.
Yeah.
Thank you very much. And related maybe specifically to antimicrobial peptides, and
Yes.
since they are naturally found as most antibiotics,
Yeah.
how can we ensure that we do not get the same problem with the resistance as we have now with the more traditional antibiotics? This
Yeah.
is obviously a very broad question, but
Of course.
what ideas are there?
Right. Antibiotics is basically, so these are compounds that are produced by bacteria or microorganisms to fight or to survive in the environment. So the concept of resistance it happens when you have a static system. But when you have a system that can evolve, I mean, actually you are forming part of this kind of race arms between, between bacteria. So you get an antibiotic. If you keep using it, eventually it will produce resistance. That's the way it is. So bacteria, because they are living organisms, right? So they have they evolve. So they have these peptides or these scaffolds that they can actually evolve to keep that race alive. So I think our scaffold is very good in this sense because it allows you to do evolution. So you can actually say, okay, I really want, and you say, well, how long it will take you to that, right? So we are developing technologies so that you can evolve or, doing, these libraries that we call to basically select for a particular loop, which amino acid is the best to target this particular target, right? So we are having very interesting results, as I was telling before, something that was not as good as for staph, very good for other gram negatives. So now doing this evolution, we can actually say, oh, wow, we are improving the activity against this. So let's say, for example, that you get some antimicrobial resistance, you could do the same. And let me say also this. So antimicrobial resistance is more, I mean, my background is as a chemist, right? So I always got intrigued in how molecules recognize each other, right? So the bigger the surface, that, like an antibody has a larger surface because it has the loops. For example, a small molecule has a smaller surface when it binds to a pocket. So my understanding is the bigger the surface, the more easiest to develop resistance because you have less, you can introduce less changes and to have in this case, resistant, the outcome the bacteria wants. You can say that. So having a larger surface, I mean, you are basically making more difficult the bacteria to develop resistance. But resistance, it will happen. It's just a question of time. And the question is how you handle that. And you use the same that nature uses. You use evolution. So evolving the system, you can actually counteract the resistance. So the question is how we can do that easily. We don't have to wait thousands of years, right? Or invest in 1,000 people in a chemical lab doing modifications. So that's the challenge. And if we have a way, so we are smart people, so we can design high throughput methods to making this, we call it positional scanning libraries, that we can actually, for example, go to one of these loops and change every amino acid and then see very quickly what is the effect and select the particular sequence that targets, for example, a particular strain or from a bacteria. So that's our approach. And I think that's kind of unique. So I haven't seen it. So that's what we are very excited about this.
And looking beyond antibiotics, what other potential uses do we have?
Some of these peptides, I mean, they are produced by not just microorganisms. I mean, in fact, we are using a protein that is produced by a mammal, right? So pig. So we have also, we call them defensins. I mean, these are peptides that have evolved in our immune system to, fight infections. So some of them are quite toxic. I mean, they're used only for local or use, in granules. So it's a challenge to use systemically this because they're challenging. I mean, they're toxic. So for example, in the PG1, it's quite toxic. One of the challenges that we are, that's one of the things that we are trying to do. So evolution can be also used. I mean, I just told you evolution can use to improve activity against the bad guys, right? But also you can put filters, say, okay, but I don't want this to be toxic to mammalian cells or to, one of the assets we use, we test activity. Is that mutation good? Or is this position good for this particular bacteria? But it's also bad for, we have a panel of mammalian cells. It's toxic to this. We also do RBC. I mean, how toxic is, for example, to red blood cells, right? How the hemolysis. That's one of some of the concerns that happens when you have peptides that are known to target bacteria membranes, right? So it can also target the good membrane. So then, so we can use this evolution to select. So these peptides, going back to your question, they have been shown, there are many papers on that they can be also, they have anti-inflammatory properties beyond the antibacterial. And when I say antibacterial, I say antimicrobial. I mean, of course, you can include antifungal, which, some microbes are fungi, and maybe antiviral. So sometimes if you have an envelope virus with a membrane and you have a peptide that somehow disrupt the membrane, you can have antiviral activity. But the most exciting is some of these peptides that actually use, for example, from mammalian systems, like they have actually anti-inflammatory properties. So which is also a very interesting property from a pharmacological point of view.
There's been not so much research yet into antimicrobial peptides. There has been, but not so much. Why do you think that is?
I mean, antimicrobial peptides, I remember when I started my PhD, I was in Barcelona, right? So, and actually I wanted my advisor, Dr. David Andreu, he's still in Barcelona. So he had a project in antimicrobial peptides. And actually that's how I got excited about this, because I remember he did a postdoc with Bruce Merrifield, which was a Nobel laureate. I mean, he discovered solid phase synthesis, right? So which has changed everything. And at that time, they were working in antibacterial peptides extracted from the hemolymph, from the blood of insects. So, I mean, it is very interesting because you see, well, you have flies flying always and eating things that are not very healthy from a human point of view, right? You know, and they don't get sick, right? So, and clearly, I mean, these insects have a very potent, it is kind of a primitive immune system. I mean, it's based on these small peptides. So the problem is that, I mean, most of these peptides are, as I said before, they have been used, for example, for topical use. I mean, you have many cases, but for systemic, it is a challenge because, I mean, these peptides, unless they are highly modified or constrained, they are not orally bioavailable, so they can be degraded by when you take it, and also they are degraded quickly by proteases, by other proteases, not just digestive proteases. So there has been a challenge, and I think, from my point of view, this has somehow put a brake on that. But now, I mean, you have all this development of new peptides. I mean, things that, like 10 years ago were not possible. Like, okay, so now we have FDA approved drugs that are peptides. So you have linaclotide, semaglutides. So I think that the second peptide that has been orally, I mean, there is what is the name? Is semaglutide, actually. So it is a GLP-1 agonist and is given orally. And so this is changing, right? So we are learning how we can make peptides better drugs. So I think this is an inflection point. I mean, now I think you're going to see more and more complex peptides to be used as antimicrobial peptides.
Julio, thanks again for your time and best of luck with that further
Yeah,
research.
thank you so much, Alistair. All right, thank you.